Every day, several sendings arrive at the Tune-In Center of Competence on the origin of timber for the identification of the processed woods. These include solid wood and wood-based materials as well as pulp and paper products. The controls of the content of the processed wood serve the implementation of the EU Timber Regulation, the protection of endangered species, and the consumer protection. The expertise is in demand of government agencies, timber trade, consumers, and associations. A paper sample is now to be examined here. The product is prepared for microscopy. For this, a small piece of paper is torn off. The first thing you do is to get a first impression of the type of product. Possibly, you will see hints that indicate that the paper contains recycled material. The paper is wetted and rolled into a ball between the fingers. Then the fibrous material is shaken up in water in order to individualize the cells. It is filtered and distributed on 2 to 3 microscope slides. Alexander Hertzberg solution is used for staining and at the same time also for embedding. First, put 2 drops of Alexander solution on the fibers and mix everything immediately with a preparation needle. Then add 1 drop of Hertzberg solution, which is also immediately mixed well with all the fibers. It is important to mix the fibers with the solution immediately after dropping on. The fibers are then evenly distributed over the surface with the help of the preparation needle and careful tapping. The cover glass is placed on top and the labeled sticker is stuck on. Now the slide can be examined under a light microscope. After the paper has been prepared, we now look at the preparation of a sample of MDF. A piece of an MDF sample is cut off with the cutter and placed in a glass beaker. The fiber material is boiled in water on the heating plate until it has dissolved. At the sink, the fibers are filtered out and rinsed under tap water. The fibers are placed on the filter and stained with nigrosine for about 10 minutes. Then they are rinsed and evenly distributed on the surface of 2 to 3 microscope slides. For embedding, 4 to 5 drops of glycerin are dripped onto them and a cover glass is placed on top. The labeled sticker is stuck on and the slide can be examined under a light microscope. For identification, we scan the slide row by row and look at the vessel elements. What does the vessel ray field look like? Are the pits all the same or do they differ? Does the vessel element have scalariform perforation plates? The standard work, Fiber Atlas by Ivesalo Fefli, was supplemented by the Atlas of Vessel Elements with important references to identify Asian timbers. Therein, you find a detailed introduction to the key features important for the identification of fibrous materials. This link allows you to download the atlas for free. For further questions, you are welcome to contact Dr. Andrea Olbrich.